Welcome everyone. I'm excited to kick off today's presentation on advanced techniques in biochemistry. Today, we'll focus on electrophoretic techniques, a crucial tool in our field. Over the next few slides, I'll guide you through the principles, methods, and applications of these techniques. Let's get started. Today, I'll be talking about electrophoresis, which is essentially the movement of charged particles when an electric field is applied. Many biological molecules like amino acids, peptides, proteins, nucleotides, and nucleic acids have ionizable groups. This means that depending on the pH of the solution, they can carry an electrical charge, either as cations, which are positively charged, or anions, which are negatively charged. When we apply an electric field, these charged particles will migrate through the medium. The direction they move in depends on their net charge. Positively charged particles, or cations, will move toward the cathode, which is the negatively charged electrode, while negatively charged particles, or anions, will head toward the anode, the positively charged electrode. Now, to perform electrophoresis, we need some basic equipment. Essentially, it consists of two main parts: a power pack, which supplies the electric current, and the electrophoresis unit itself, where the actual separation takes place. These components work together to allow us to analyze and separate biological molecules effectively. Now, let's talk about the types of electrophoresis units available for running gel systems. These units can be configured as either vertical or horizontal setups, depending on the specific application. In particular, vertical slab gel units are very common and are routinely used to separate proteins within acrylamide gels. The key here is that the gel is formed between two glass plates, which are clamped together to hold the gel in place. To create the space needed for the gel, plastic spacers are used, ensuring the gel has the correct thickness. When it comes to popular choices, the most widely used units are what we call the Minigel apparatus. These are compact, convenient, and ideal for many laboratory workflows, making them a staple in protein analysis. Understanding these units is essential because they form the backbone of many electrophoretic separation techniques, helping us analyze proteins accurately and efficiently. Now, let's look at how we prepare the gel for electrophoresis. First, a plastic comb is placed into the gel solution before it polymerizes. Once the gel has set, we remove the comb, which creates loading wells. These are where you'll load up to 10 samples for analysis. It's a simple but crucial step because these wells are where the samples will be introduced into the gel matrix. When we assemble the electrophoresis apparatus, the lower tank is filled with buffer solution that surrounds the gel plates. This buffer isn't just there for conductivity. It also helps keep the gel cool during the run, which is important for maintaining consistent results. The gel itself is typically cast on a glass or plastic sheet and then placed on a cooling plate. This cooling plate is an insulated surface through which water is circulated to absorb and carry away heat generated during electrophoresis. Managing heat is vital because excess heat can distort the gel or affect the separation process. Finally, we connect everything to the power pack, which supplies a direct current across the electrodes in the electrophoresis unit. This current drives the movement of charged molecules through the gel, enabling us to analyze their sizes and properties effectively. All these steps, casting the gel, setting up the buffer system, and connecting the power, work together to ensure a smooth, controlled separation process. This setup is fundamental in the broader context of molecular analysis techniques we're exploring today. All electrophoresis is carried out in an appropriate buffer, which is essential to maintain a constant state of ionization of the molecules being separated. This means that the buffer helps keep the molecules' charge stable during the process. If the pH level of the buffer changes, it can affect the overall charge of the molecules. And as we know, the charge directly influences how fast or slow these molecules move through the medium. So, maintaining a consistent pH is crucial because it ensures that the mobilities of the molecules remain predictable and reliable. This consistency is key to achieving accurate separation results. It ties into the broader theme of electrophoresis being a precise and controlled technique, where even small changes in conditions can significantly impact the outcome. Let's take a closer look at a typical horizontal electrophoretic apparatus. As you can see, this setup is designed to separate molecules, usually proteins or nucleic acids, based on their size and charge. The key components include the gel chamber, where the actual separation happens, and the power supply that creates an electric field across the gel. The gel itself is usually made of agarose or polyacrylamide, depending on what we're analyzing. When we apply an electric current, the molecules migrate through the gel matrix. Smaller molecules tend to move faster and travel farther, while larger ones move more slowly. This setup is quite efficient and commonly used because it allows easy visualization and extraction of separated molecules. It's a fundamental tool in molecular biology labs, connecting directly to our overall theme of understanding molecular structures and functions through separation techniques. So, this apparatus is essentially the workhorse that helps us analyze complex biological samples by giving us a clear, visual way to distinguish different molecules. The introduction of gels as a support medium really marked a turning point in how we analyze macromolecules. It allowed for much more precise and efficient separation techniques. The very first gel system used was starch gel, which played a foundational role. Although starch gels are still used in some applications today, most modern electrophoretic methods now rely on either agarose gels or polyacrylamide gels. These gels have become the standard because they offer better resolution and are more versatile for different types of analyses. Overall, the shift to gel-based support media significantly advanced our ability to study complex biological molecules. Agarose is a linear polysaccharide, which means it's made up of long chains of sugar molecules arranged in a straight line. The basic building block of agarose is called agarose, a repeating unit that consists of 2 different sugars, galactose and 3,6-anhydrogalactose. These sugars alternate along the chain, creating a specific structure that's essential for its properties. Agarose is actually one of the key components of agar, the substance we often use in microbiology. Agar is a mixture of different polysaccharides, and agarose makes up a significant part of it. When we prepare agarose for gel electrophoresis, we typically use concentrations between 1% and 3%. This range provides the right consistency for the gels to support the separation of molecules like DNA. To make an agarose gel, we suspend the dry agarose powder in an aqueous buffer, basically a water-based solution. Then, we boil this mixture until it becomes clear, indicating that the agarose has dissolved completely. Once it's clear, we pour the hot solution into a mold and let it cool down to room temperature. As it cools, it solidifies into a rigid gel, which is perfect for running electrophoresis experiments. So, understanding how agarose forms and behaves is fundamental to its role in molecular biology techniques. Let's take a closer look at the agarose casting tray. This is a crucial component in gel electrophoresis because it's what we use to shape our gel. Essentially, the tray holds the molten agarose as it solidifies into a gel matrix. It's designed to create a uniform, flat surface, which is key for consistent DNA migration during the run. The tray typically consists of a base with a well or multiple wells at one end, where we pour the agarose solution. Once it cools and solidifies, we have a stable platform for loading our samples. The design also usually includes a comb to form wells in the gel, where we'll load our DNA samples later on. Connecting this to the overall process, the agarose casting tray helps ensure that the gel is properly formed and ready for electrophoresis. Its role might seem simple, but it's essential for achieving clear, reliable results in DNA analysis. So, choosing the right tray and preparing it correctly sets the foundation for successful experiments. This slide doesn't contain enough information. That's why we're moving to the next slide. Agarose gels are a versatile tool used for the electrophoresis of both proteins and nucleic acids. When it comes to proteins, the pore sizes in a 1% agarose gel are quite large compared to the size of the proteins themselves. This means that proteins can move freely through the gel matrix without being hindered by small pores. Because of this property, agarose gels are especially useful in techniques like flatbed isoelectric focusing, where we want proteins to migrate according to their native charge. In this setup, the large pore size allows proteins to move unimpeded, helping us analyze their charge properties more effectively. Similarly, these large pore gels are ideal for separating much larger molecules, such as DNA or RNA. The pores are still big enough for these nucleic acids to pass through easily. So, whether we're working with proteins or nucleic acids, agarose gels provide a flexible and effective medium for separation based on size or charge. This highlights their importance in various molecular biology applications and emphasizes their role in our overall understanding of biomolecular analysis. Electrophoresis in acrylamide gels is often called PAGE, which stands for Polyacrylamide Gel Electrophoresis. It's a common technique used to separate proteins based on size and charge. Now, these gels are formed through a process called polymerization, where acrylamide monomers link together to form long chains. To create a stable gel, we add a small amount of N,N-methylene bisacrylamide, or bisacrylamide, which acts as a cross-linker. Think of bisacrylamide as two acrylamide molecules connected by a methylene group, essentially bridging the chains together. During polymerization, acrylamide monomers polymerize in a head-to-tail fashion, forming long, linear chains. Occasionally, a bisacrylamide molecule gets incorporated into the growing chain, creating a second point where the chain can extend, which helps form a stable, three-dimensional network. This polymerization process is a free radical catalysis, meaning it's initiated by free radicals. The radicals are generated by adding ammonium persulfate, or APS, and TEMED, which is N,N,N-tetramethylnadiamine. These initiators kickstart the chain reaction, leading to the formation of the gel matrix. In essence, this controlled polymerization creates a mesh-like structure that's perfect for separating molecules during electrophoresis, especially proteins, with high resolution. Understanding this process helps us grasp how PAGE works and why it's such a powerful tool in biochemistry. Cross-linked polyacrylamide gels are formed through a process called polymerization, which involves acrylamide monomers. Essentially, these monomers join together to create a network, and the cross-linking points help connect different polymer chains. This structure is crucial because it gives the gel its stability and porosity, making it ideal for separating biomolecules like proteins and nucleic acids. Throughout this presentation, we've seen how the properties of these gels influence their function in various applications, especially in electrophoresis. So, understanding how these gels are formed from acrylamide monomers helps us appreciate their versatility and effectiveness in laboratory techniques. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Today, we're going to explore the fascinating process of electrophoresis of proteins. This technique is fundamental in biochemistry because it allows us to separate and analyze proteins based on their size and charge. Essentially, when we apply an electric current to a gel containing proteins, these molecules migrate through the gel matrix at different rates. The key idea here is that small or highly charged proteins move faster, while larger or less charged ones move more slowly. This separation helps us identify specific proteins within complex mixtures. Electrophoresis is a vital tool in many applications, from diagnosing diseases to purifying proteins for research. By understanding how proteins behave under an electric field, we can glean a lot of information about their properties and functions. So, this slide highlights the importance of electrophoresis as a cornerstone technique in protein analysis, tying into our broader theme of understanding protein structure and function. Today, I want to talk about SDS, Polyacrylamide Gel Electrophoresis, or SDS-PAGE, which is actually the most commonly used method for analyzing protein mixtures qualitatively. It's especially useful when we want to monitor protein purification processes. Since this method separates proteins based on their size, It can also help us determine the relative molecular weight of different proteins. When preparing samples for SDS-PAGE, the first step is to boil them for about 5 minutes in a sample buffer that contains beta-mercaptothenol and SDS. Boiling helps denature the proteins, making them linear. The beta-mercaptothenol plays a crucial role by reducing any disulfide bonds that might be holding the protein's tertiary structure together. Meanwhile, SDS binds strongly to the proteins, unfolding and denaturing them completely. This ensures that the separation we see on the gel is solely based on size, not shape or charge. In essence, SDS-PAGE is a powerful, straightforward technique that allows us to analyze proteins efficiently and accurately, which ties directly into our broader goal of understanding protein structure and function. Today, I'll be explaining the principle behind SDS-PAGE, which is a fundamental technique in protein analysis. SDS-PAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. The key idea here is that we're separating proteins based on their size or molecular weight. To do this effectively, we first treat the proteins with SDS, which is an anionic detergent. This detergent binds uniformly along the length of the proteins, giving them a consistent negative charge proportional to their size. This uniform coating is crucial because it ensures that when we run the gel, the protein's movement is primarily influenced by their size, not their charge or shape. The polyacrylamide gel itself acts as a molecular sieve, slowing down larger proteins more than smaller ones. As an electric current is applied, the negatively charged proteins migrate through the gel matrix towards the positive electrode. Smaller proteins move faster and therefore travel farther, allowing us to separate a mixture of proteins effectively. This technique is essential because it helps us determine the size of unknown proteins, compare different samples, and analyze protein purity. Overall, SDS-PAGE is a powerful, widely used method that hinges on the principles of uniform charge coding and size-based separation, tying into our broader goal of understanding protein structure and function. Now, let's look at what happens to each protein in the mixture during this process. When they are treated with SDS, they become fully denatured, meaning they lose their original three-dimensional structure. They then open up into a rod-shaped form, with a series of negatively charged SDS molecules attached along the entire length of the polypeptide chain. On average, one SDS molecule binds for every 2 amino acid residues. This is a key point because it shows how SDS effectively coats the protein, giving it a uniform negative charge. As a result, the original native charge of the protein is completely overshadowed by these negatively charged SDS molecules. This rod-like shape remains stable because if the protein tries to fold back into a more compact form, the negative charges on different parts of the chain would repel each other. This electrostatic repulsion prevents the protein from folding up again, forcing it to stay in that extended, rod-like conformation. And this is fundamental to SDS, polyacrylamide gel electrophoresis, because it allows us to separate proteins based on size rather than shape or charge. The uniform negative charge along the extended protein ensures that when an electric field is applied, the proteins migrate through the gel primarily according to their length. Typically, the separating gel used in electrophoresis is a 15% polyacrylamide gel. This type of gel creates a specific pore size that influences how proteins move through it. Proteins with a relative molecular mass, or MR, of around 10,000 tend to move quite easily, or relatively unhindered, through this gel. On the other hand, proteins with a MR of about 100,000 can just barely enter the pores of a 15% gel. This means that gels with 15% polyacrylamide are particularly useful for separating proteins within the range of about 10,000 to 100,000 in MR. But if you have a protein that's larger, say around 150,000 MR, it would be unable to enter a 15% gel at all. In such cases, you'd opt for a gel with larger pores, like a 10% or even a 7.5% gel, so that the bigger protein can enter and be stained or identified properly. It's clear, then, that choosing the right gel depends heavily on the size of the protein you're studying. The fractionation range of different percentage acrylamide gels is summarized in Table 10.1, which shows, for example, that a 10% gel can't let proteins larger than 200 kDa enter, but can separate proteins ranging from about 15,000 to 200,000 in Mr. Proteins of 15,000 Mr. or less are too small to be effectively sieved by the gel matrix, so they all run together as a single band at the electrophoresis front. Understanding these principles helps us select the appropriate gel for our specific protein analysis, ensuring accurate separation and identification. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at the SDS-PAGE gel. This technique is fundamental in protein analysis because it allows us to separate proteins based on their size. The gel itself acts like a molecular sieve, where smaller proteins migrate faster through the matrix, while larger ones move more slowly. By applying an electric current, we cause the negatively charged proteins to travel through the gel toward the positive electrode. This separation process is crucial for identifying and characterizing proteins in complex mixtures. Throughout our presentation, we've seen how SDS-PAGE helps us visualize protein purity, estimate molecular weight, and assess sample quality. It's a powerful tool that underpins many of our downstream applications, making it an essential step in protein research. While SDS-PAGE is the most commonly used gel system for studying proteins, it has limitations when it comes to detecting a specific protein based on its biological activity. That's because the SDS-PAGE process denatures the proteins, including enzymes, rendering them inactive. So, if you want to identify an enzyme by its activity, you need to use non-denaturing conditions. In this case, we work with native or buffer gels. These are typically polyacrylamide gels, usually around 7.5%, but crucially, the SDS is absent. This means the proteins are not denatured before loading onto the gel. Since the proteins retain their native charge at the gel's pH, which is usually around 8.7, they separate based on their electrophoretic mobility and the sieving effects of the gel. It's important to note that predicting how a specific protein will behave in a buffer gel can be tricky because proteins have different sizes and charges. However, this diversity actually helps us achieve good resolution across a mixture of proteins. To identify an enzyme of interest in such a gel, we can incubate it with a suitable substrate. If the enzyme is active, it will produce a colored product right at its location in the gel, making detection straightforward. Alternatively, we can include the substrate in an agarose gel poured over the acrylamide gel. As the enzyme diffuses and interacts with the substrate between the two gels, a color will develop at the enzyme site, confirming its activity. So, working with native gels allows us to preserve enzyme activity and directly observe biological function, which is a key aspect of studying proteins in their active forms. This is again a polyacrylamide gel system, but instead of running a slab gel of uniform pore size throughout, such as a 15% gel, we have a gradient gel. In this case, the acrylamide concentration varies uniformly from, typically, 5% at the top to 25% at the bottom of the gel. The gradient is formed using a gradient mixer and is run down between the glass plates of a slab gel. The higher percentage acrylamide, like 25%, is poured between the glass plates first, and then a continuous gradient of decreasing acrylamide concentration follows. This means that at the top of the gel, there are large pores due to the 5% acrylamide. But as the sample moves down through the gel, the acrylamide concentration slowly increases, and the pore size correspondingly decreases. Gradient gels are usually run as SDS gels with a stacking gel. There are two main advantages to using gradient gels. First, they allow us to separate a much broader range of protein molecular weights than a fixed percentage gel. This makes them incredibly versatile for analyzing complex protein mixtures. Today, I want to talk about gradient gels. These are a really useful tool in the lab because they allow us to analyze protein mixtures with high precision. Essentially, gradient gels have a varying concentration of acrylamide, which creates a continuous range of pore sizes. This means that smaller proteins can move more easily through the gel, while larger proteins are separated at different rates depending on their size. This gradual change in pore size improves the resolution of protein separation, especially when we're dealing with proteins that are similar in size. It's a key technique for achieving clear, distinct bands in our electrophoresis results. In the context of our overall presentation, gradient gels exemplify how fine-tuning experimental conditions can lead to more accurate and insightful data. They highlight the importance of optimizing separation methods to better understand complex protein mixtures. This method is ideal for the separation of amphoteric substances, such as proteins, because it relies on the different isoelectric points of molecules. What that means is, it separates proteins based on the pH at which they carry no net charge, which is their isoelectric point. This allows for very precise separation, especially since this technique can distinguish proteins that differ in their isoelectric points by as little as 0.01 pH units. The most common system used for isoelectric focusing, or IEF, involves horizontal gels that are placed on glass plates or plastic sheets. The separation occurs by applying a voltage across the gel, which contains a carefully established pH gradient. Now, creating that pH gradient is a key part of the process. It's achieved by introducing compounds called ampholites into the gel. Ampholites are complex mixtures of synthetic polyaminopolycarboxylic acids, and they help establish a stable and continuous pH gradient across the gel. And these gels, known as isoelectric focusing gels, are specially designed to facilitate this separation. Overall, this technique offers high resolution and is particularly effective for analyzing complex mixtures of proteins, connecting directly to our theme of advanced separation methods. This slide doesn't contain enough information. That's why we're moving to the next slide. The most commonly used general protein stain for detecting protein on gels is the sulfated trimethylamine dicoumacy brilliant blue R250, or CBB for short. When we stain, we typically use a solution that's about 0.1% weight volume of CBB in a mixture of methanol, water, and glacial acetic acid, in a ratio of 45:10 by volume. That acid methanol mixture acts as a denaturant, which helps to fix the protein in the gel. This step is crucial because it prevents the protein from washing out while it's being stained. Usually, staining takes around 2 hours, and then we do the destaining, which is often overnight. During destaining, we gently agitate the gel in the same solution but without the dye, which helps to remove excess stain and increase the contrast of the protein bands. Now, CBB is highly sensitive, meaning that even a very weakly stained band on a polyacrylamide gel can correspond to about 0.1 milligrams or 100 nanograms of protein. However, it's important to note that CBB isn't used for staining cellulose acetate membranes or protein blots because it binds strongly to the paper. In those cases, proteins are first denatured by briefly immersing the strip in 10% trichloroacetic acid, and then stained with a dye that doesn't stain the support material, like Prussian blue, Amido black, or Prussian S. Although CBB is quite sensitive, some researchers need even greater sensitivity. That's where silver staining comes in. Silver stains are based either on techniques originally developed for histology or on photographic methods. The principle is that silver ions are reduced to metallic silver on the protein, depositing as a black or brown band. Silver staining can be performed right after electrophoresis or after staining with CBB, providing flexibility depending on the experimental needs. In terms of detection, estimation, and recovery of proteins from gels, these staining methods are essential tools. They allow us to visualize proteins clearly, estimate their quantities, and even recover them for further analysis. This entire process ties back to our overall goal of understanding protein presence and abundance in different samples, which is fundamental in many biological studies. This slide doesn't contain enough information. That's why we're moving to the next slide. Although PAGE is primarily an analytical technique, it also has the ability to fractionate a protein mixture during electrophoresis. This means that after separation, we can actually take individual proteins and examine them more closely. The first step in this process is to transfer or blot the separated proteins from the gel onto a solid support, usually a sheet of nitrocellulose paper. This process is called protein blotting, or more specifically, Western blotting, which is named by analogy to Southern blotting used for DNA. The transfer of proteins from the gel onto the nitrocellulose is achieved through a method called electroblotting. In this technique, we create a sort of sandwich with the gel and the nitrocellulose, place it in a cassette, and then immerse it in buffer between two parallel electrodes. When we pass an electric current at right angles to the gel, it causes the separated proteins to migrate out of the gel and onto the nitrocellulose sheet. Once this transfer is complete, the nitrocellulose with the proteins attached is referred to as a blot. This blot can then be further examined, often by probing it with specific antibodies to detect particular proteins. This step allows us to identify and analyze proteins of interest with high specificity, making Western blotting a powerful tool in protein analysis. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about electrophoresis of nucleic acids. This technique is fundamental in molecular biology for separating DNA or RNA molecules based on their size. The process involves applying an electric current to a gel matrix, typically agarose or polyacrylamide. Because nucleic acids are negatively charged, they migrate towards the positive electrode when an electric field is applied. The key point here is that smaller fragments move faster and travel farther through the gel, allowing us to distinguish between different sizes of nucleic acids. This separation is crucial for various applications, such as analyzing PCR products, checking DNA purity, or preparing samples for further experiments. Connecting this to the overall theme, electrophoresis provides a visual and quantitative way to analyze nucleic acid molecules, helping us understand their structure, size, and integrity. It's a powerful tool that underpins much of modern genetic research and diagnostics. For most DNA samples, we typically use agarose gels for electrophoretic separation. This is mainly because the DNA molecules and fragments we analyze regularly tend to be much larger than proteins. The key point here is that larger DNA fragments can't easily enter a polyacrylamide gel due to its smaller pore size. So, agarose gels, with their larger pores, are more suitable for this purpose. Since the charge per unit length in DNA is consistent, thanks to the phosphate groups, any given DNA fragment will move toward the anode at a consistent rate when an electrical field is applied. This uniform charge-to-mass ratio is what allows us to compare DNA fragments based on their migration distance through the gel. And when we look at agarose gel electrophoresis of DNA, it's a straightforward and effective way to separate these molecules by size, which is essential for many genetic analyses. Overall, understanding why we choose agarose for DNA helps us appreciate how the physical properties of DNA influence our lab techniques. So, in agarose gels, separation of molecules happens because the gel creates resistance to their movement. Think of it like a filter. The gel matrix slows down molecules as they try to pass through. The larger molecules, like very big DNA fragments, will have the hardest time moving through these tiny pores, and in some cases, they might even get completely blocked. On the other hand, the small molecules can navigate through the gel more easily, so they move relatively unhindered. This means that during gel electrophoresis, the mobility of DNA molecules depends primarily on their size. The smaller the DNA, the faster it moves. Now, this is somewhat similar to how proteins separate in SDS polyacrylamide gels, but it's not a perfect analogy. Unlike proteins, double-stranded DNA tends to form quite stiff rods, which influences how they pass through the gel. It's believed that long DNA molecules pass through the gel pores end-on, rather than threading through like spaghetti. As DNA moves through the pores, it experiences drag, basically, resistance, and the longer the molecule, the more drag it encounters. This means longer DNA fragments are slowed down more than shorter ones. For very small double-stranded DNA, sideways movement may become more significant, as they can shift side to side more easily within the gel. Overall, Agarose Gel Electrophoresis allows us to separate DNA based on size, and understanding how the molecules interact with the gel matrix helps us interpret the results more accurately. Like that of DNA, electrophoresis of RNA is usually carried out in agarose gels, and the principle of the separation, based on size, is the same. Often, we need a quick way to check the integrity of RNA right after extracting it, before deciding whether to proceed with further analysis. This can be easily achieved by running an electrophoresis in a 2% agarose gel, which typically takes about an hour. When we do this, you'll see that the ribosomal RNAs, specifically the 18S and 28S bands, are quite clear and easy to identify. This step is crucial because it allows us to verify that our RNA is intact and suitable for downstream applications. So, in essence, electrophoresis provides a quick, reliable snapshot of RNA quality, helping us make informed decisions early in our workflow. The technique we've been discussing has gone by several names, including high-performance capillary electrophoresis, or HPCE, capillary zone electrophoresis, CZE, free solution capillary electrophoresis, FSCE, and simply capillary electrophoresis, CE. Nowadays, the most common term you'll hear is CE. This method is notable for its microscale nature. The capillaries used are very small, which means only microliters of reagents are needed for analysis, and just nanoliters of sample are sufficient. This tiny scale not only saves on materials, but also allows for very sensitive detection, sometimes down to femtomole levels. That's 10 to the power of minus 15 moles. Because of these features, capillary electrophoresis has become a preferred method in many biomedical and clinical settings. Its high sensitivity and minimal sample requirements make it ideal for analyzing a wide variety of biological molecules. You can separate amino acids, peptides, proteins, DNA fragments like synthetic oligonucleotides, and other nucleic acids. It's also effective for small organic molecules, such as drugs or even metal ions. Overall, capillary electrophoresis is a powerful, versatile tool that provides efficient separation and detection, making it invaluable in many research and diagnostic applications. This slide doesn't contain enough information. That's why we're moving to the next slide. The further miniaturization of electrophoretic systems has led to the development of microchip electrophoresis, which offers many advantages over traditional methods. It allows for very high-speed analyses, and it requires only tiny sample sizes. For example, with microchip analysis, you can often complete a run in just tens of seconds. In contrast, capillary electrophoresis, or CE, can take around 20 minutes, and conventional gel electrophoresis can take at least 2 hours. One of the key innovations here is the use of new detection systems, such as laser-induced fluorescence. This technology can achieve sensitivity levels from picomoles down to attomoles, which is an astounding 10 to the minus 18 moles, at least 100 times more sensitive than conventional CE. When it comes to molecules that don't fluoresce naturally, we have other detection options, like electrochemical detectors, pulsed amperometric detection, or sinusoidal voltammetry. These methods all provide high sensitivity, are very well suited to miniaturization, and tend to be low cost. They are also highly compatible with advanced microfabrication technologies, which allows for further integration and miniaturization of the systems. Finally, an important benefit of microchip electrophoresis is that it only requires a few volts of applied voltage, unlike traditional CE systems that need high voltages. This makes the whole process safer, more portable, and easier to implement in various settings. Microchip electrophoresis, or MCE, is essentially a miniaturized version of the traditional capillary electrophoresis technique. As many of you know, electrophoresis is a widely used method to separate large molecules like nucleic acids, DNA and RNA, and proteins. It's become a routine tool in labs worldwide for tasks such as sizing DNA fragments or separating complex protein mixtures. What makes MCE particularly exciting is that by applying higher voltages, we can achieve faster and more efficient separations. This is one of the key advantages of miniaturizing the system. Now, the process of creating these microchips is called microfabrication. It involves etching very precise, reproducible channels, think of tiny capillaries, on surfaces made of quartz, glass, or plastic. These channels are about 50 micrometers wide and 10 micrometers deep, which is just a bit smaller than a human hair. Once the channels are etched, a second sheet is fused on top, sealing them into closed microfluidic pathways. These channels connect to reservoirs that allow us to introduce or remove fluids easily. The entire chip is remarkably small, often just a couple of square centimeters in size. Overall, what makes microchip electrophoresis so compelling is that it offers a system similar to traditional capillary electrophoresis, but with much more flexibility. It's faster, more adaptable, and opens up new possibilities for miniaturized, high-throughput analysis. This slide doesn't contain enough information. That's why we're moving to the next slide.